The overall goal of this procedure is to advance our capacity to protect individuals with antibodies or vaccines for preventing or treating histoplasmosis, and to examine the role of virulence factors in disease pathogenesis. This is accomplished by first isolating and characterizing monoclonal antibodies to target antigens. The second step of the procedure is to administer the selected monoclonal antibodies intraperitoneally 2 hours prior to the intranasal infection, in order for these molecules to have time to distribute throughout the body. The third step of the procedure is to anesthetize the mice and intranasally infect them with the desired inoculum. The final step of the procedure is to monitor the health of the animals. The animals are either euthanized at various intervals to determine fungal burdens and to assess inflammatory responses, or are followed for survival. Ultimately, results can be obtained that show a prolongation in survival and a modification of pathological responses in the affected organs of lethally infected mice through peritoneal administration of the protective monoclonal antibodies prior to infection.
Demonstrating the procedure will be Allan Guimarães, a postdoctoral student with extensive experience in fungal pathogenesis and animal infection models.
The main advantage of this technique over existing methods like antifungal drugs is that the monoclonal antibodies are effector molecules with fungicidal properties that work through engagement of the host's potential effector response to disease. Visual demonstration of this method is critical, as the monoclonal antibody administration and intranasal infection steps are difficult to learn because they require expertise in handling the animals.